So welcome everybody for this new episode of the Forefront of Virology COVID-19 webinar series. And actually today we will not be talking about COVID-19 because these webinars are really meant the more and more to really expand, and we will change the titles to address all human viruses. And this is really the spirit of the Global Virus Network. As always, because maybe some of you do not really know what the Global Virus Network is about, just very few words. This is a network, science-driven, independent, now merging around 70 research centers worldwide and 12 affiliates. And the mission of this network, which has been created by Bob Gallo and Billy Hall in 2011, is really the missions are really three of them: research, in particular through what we call task force on different topics, education, very important, and training with the newly created GVN Academy. We provide fellowships, we really nurture the career tracks of young investigators with the so-called Rising Star program and so on, and communication and advocacy. It's about expertise, it's about partnerships with national and international institutions, with academic institutions, and with industrial partners, with in particular what we call the GVN Corporate Center partners, which is an important component now of the GVN. And in all this context, these webinars are very important to provide information, of course, to gather the community. And this has been increasingly important with very important webinars over the past few months and years. So in this context, it's really my pleasure to welcome Stephan Becker today. Stephan has been educated in Germany. He actually got his postdoc, and then he became an assistant professor and an associate professor at the Philipps Universität Marburg. And then he moved from 2005 to 2007 as becoming the head of the P4 program at the Robert Koch Institute in Berlin, and then went back to Marburg, and he's now professor of virology and director of the Institute for Virology at the Philipps Universität in Marburg. Stephan is heavily involved in many programs, in many collaborative programs. He's a member, many of them on pandemics, on emerging pathogens. He's a member of high-level scientific societies and communities, and notably he's since 2015 a member of the National Academy of Science Leopoldina. He's the author of over 249 publications with a very high H index of 81. Stephan is really an expert, and he's also the director of the GVN Center. And so we really very much welcome his lecture on Ebola, which illustrates again that the GVN is dealing with all human viruses. And we look forward, Stephan, to your lecture. Thank you so much.
Thank you very much, Christian, for the invitation, first of all, and for the nice introduction. Yes, we are working with mainly with Ebola virus in my group, but also with Marburg virus. And now I will try to share the screen. It worked once already. So
yes, we can see it.
You see. Okay, now today I wanted to share some data about post-translational modifications of Marburg, of Ebola virus proteins, and moreover the effects those post-translational modifications have on Ebola virus transcription and budding. So just to introduce the way filoviruses made it to Marburg, which is a little city in in the center of Germany and has nothing to do with Africa so far, at least not at this time. And we are going back to the year 1967. And this is the local newspaper of the city of Marburg. And the title in German is Mysteriöse Erkrankung von 11 Angestellten. And in English, this would be Mysterious Disease of 11 Employees. And this mysterious disease was actually a hemorrhagic fever. And in this time in 1967, it was really something very special. This was the first time that such a hemorrhagic fever appeared in the city of Marburg. And so the outbreak that appeared at this time was mainly in Marburg, but also there were cases in Frankfurt and in Belgrade. And the primary cases that came down with the hemorrhagic fever were lab workers, which had contact to tissue of imported monkeys. And these imported monkeys came from Uganda. And actually, you might imagine what it meant to Marburg. This is really a little city with, at this time, maybe 60,000 inhabitants. When such a severe disease appears, and this hemorrhagic fever was really something special to all of them, and everybody was really frightened and afraid that he or she may be the next one which will develop the disease, because nobody really knew what kind of disease this was. It was not clear how this is transmitted, and therefore the whole city was really in a very special situation. Then after 2 weeks approximately, then it became clear that the disease is not transmitted via via aerosols or airborne. So the transmission needed a very close contact to a patient or to the tissue of one of these obviously infected monkeys. And then there were secondary cases, only a few, and these were relatives or healthcare workers. So fortunately, this was a quite small outbreak of 32 cases and with a case fatality rate of about 25%. But still, nobody knew what this outbreak, what was the causative agent of this outbreak. And then 2 people in Marburg, Werner Slenska and Rudolf Siegert, 2 virologists, they started to look into the cause of the disease. And after 3 months of feverish work, if you wish, they came up with an electron micrograph of a virus, which looked like this. And this virus was of a morphology that nobody ever seen before, filamentous virus, which sometimes was bent into 6 shape or like you see here on this picture. And this was then found to be the causative agent of this hemorrhagic fever. And this virus was called Marburg virus. And at this time, it was not, it was really a discussion whether this should be called Marburg virus, but not so that the people in Marburg or the virologists were afraid of that this is called Marburg virus, but everybody wanted to have this virus named after the city, because there are not only these 2 guys here from Marburg who were involved in the outbreak investigations, but many other international colleagues. And for example, people from Frankfurt and from Freiburg, and everybody wanted to have this new virus called according to his own home city. This is different now. So this, anyway, this outbreak in 1967 started the work with emerging viruses in Marburg, and now we do this work in this BSL-4 facility that you can see here. And we work under full protection, as you can see here. This is Anke Werner, one of the students who did a lot of the work that I will present today. So after this 1967 outbreak, there was then, after this was stopped, nobody knew again anything about Marburg virus. And for 9 years, this whole story about this hemorrhagic fever virus were silent. And then in 1976, Ebola virus was detected in Sudan and Zaire. And the electron micrographs that have been performed at this time looked very similar to the morphology of the Marburg virus, and it became clear that Ebola virus and Marburg virus belong to the same family, and this was called Filoviridae. And since then, there were sporadic outbreaks in Africa, and it seems that during the last years, as you can see here in this graph, that during the last years, the outbreaks started to become more frequent, and the largest outbreak was in 2014, 2015 in Western Africa, with close to 30,000 cases and 11,000 case fatalities. So this was an Ebola virus Zaire outbreak in Western Africa. Then we had additional outbreaks in Africa. This was a Democratic Republic of Congo, a large outbreak. And then 2020, another Ebola virus outbreak. And you all know that there has been an outbreak of Sudan Ebola virus in Uganda. So as I said before, the transmission of Ebola virus and also of Marburg virus is through body fluids and close contact. The symptoms is hemorrhagic fever, diarrhea, and the case fatality rate is depending on the virus and on the site of the outbreak between 25 and 88%. So the latest outbreak of Sudan Ebola virus in Uganda was declared over a couple of days ago, meaning that there are always outbreaks of Filoviridae, of Ebola virus, of Marburg virus in Central Africa. And I think we can be sure that the next outbreak is already in sight. So today it's about molecular biology of Ebola virus. And I therefore would like to present you some molecular characteristic of the virus. As you can see here, this is Ebola virus in an electron micrograph. The genome of the Ebola virus is a single-stranded negative sensed RNA. There are 7 consecutive genes on this RNA genome, encoding for 8 or 9 different proteins. From the glycoprotein, there are several forms that are made. And this is a schematic of the virus now. What you can see here is the surface. And in the middle there, in the center of this particle, there is the nucleocapsid, which is composed of the nucleoprotein, VP35, VP24, the L protein, and VP30. And VP30 is the protein I will talk about this morning. So the nucleoprotein is important for the genome encapsulation. As you can see here, the nucleoprotein is the major factor that forms the nucleocapsid. And then we have a complex of the large polymerase protein and a cofactor of the polymerase VP35, which together make the polymerase function of Ebola virus. And then we have VP30, which, as I said, I talked about, and VP24. This is a protein that seems to induce the final form of the nucleocapsids before the nucleocapsids can be transported. Then we have 2 additional proteins, VP40, which is the budding function of Ebola virus, and then the glycoprotein GP, which is the surface protein and the receptor recognition protein of Ebola virus. So this is, okay. Now, concerning the Ebola virus replication cycle, it starts with the attachment of the particle at the plasma membrane of the target cell, and then via macropinocytosis, these particles are engulfed, and then finally in the late endosome or lysosome, the membrane of the virus fuses with the membrane of the endosome, and with this fusion, the nucleocapsid is released into the cytoplasm and there serves as a template for transcription first. And the first function that we see from the polymerase is to do primary transcription synthesizing the mRNAs of Ebola virus, which then are translated by the cellular translational machinery. And as soon as the viral proteins appear then in the cytosol, what we see then is the formation of inclusion bodies. And the inclusion bodies are formed by liquid-liquid phase separation when the nucleoprotein itself is appearing inside of the cell. And then the nucleoprotein recruits all the other nucleocapsid proteins and also VP40 into these inclusion bodies. And these inclusion bodies are also the sites of secondary transcription, but also genome replication. And as soon as the genome is replicated, it is encapsulated by the nucleoprotein. The nucleoprotein then recruits all the other nucleocapsid proteins, and then the mature nucleocapsids are transported in an actin-driven process to the plasma membrane. And there are mainly two sites where filopodia are formed. This is a cellular protrusion. And these nucleocapsids then make their way into the filopodia and then are released as infectious particles into the supernatant. Okay, so this is the replication cycle of Ebola virus. Now, the question of this presentation is mainly, how does Ebola virus regulate transcription and replication, viral transcription and viral replication? So for this, I would start with some information which we found out quite a long time ago. And this was that the nucleocapsid proteins of filoviruses, of Ebola virus and of Marburg virus, they form protein complexes. So the nucleoprotein is able to interact with VP35, and the nucleoprotein is also able to interact with VP30. And by these interactions, the nucleoprotein recruits all the other nucleocapsid proteins into these inclusions that you can see here in this immunofluorescence pictures. So what we found out so far was that the nucleoprotein is the central player in recruiting all the other nucleocapsid proteins into these inclusion bodies where replication and transcription takes place. And therefore, of course, all the proteins that are involved in these processes are assembled in the inclusions. So then we also, very long time ago, we established a mini replicon system for filoviruses, and this we needed to identify the different proteins and their role in this, in replication and transcription. And what you can see here, if we look for replication, we found that there were three nucleocapsid proteins essential to synthesize the genomic RNA. And this was the polymerase and the cofactor of the polymerase and the nucleoprotein which encapsulated the RNA. And when we then added VP30, it does not seem to make a big difference. This was very different with transcription. As you can see here, the viral mRNA, which is formed by this mini replicon system, is only seen if we have four proteins together, L, VP35, and NP, as for replication. And then we need also VP30, and only in the presence of VP30, the mRNA of Ebola virus or Marburg virus can be made. So what we learned from this was that VP30 was an essential Ebola virus transcription factor. So three proteins were necessary for replication, NP, VP35, and L, and four proteins were necessary for transcription. And the difference was the presence of VP30. Only in the presence of VP30 transcription could take place with Ebola virus, for Ebola virus. Then we were looking at this time also at post-translational modifications, and especially for phosphorylation of the nucleocapsid proteins of Marburg virus here in this case. And we found that there were three of the nucleocapsid proteins, the nucleoprotein and VP30 and VP35. There's only a very faint band. I don't know whether you can see this. these three proteins were phosphorylated. The same was also true for Ebola virus nucleocapsid proteins. And the phosphorylation of VP30 is the center of this talk. And as you can see here, so VP30, obviously the transcription factor, was phosphorylated by a so far unknown cellular kinase. So we were then, of course, interested in the phosphorylation of VP30 because at this time nothing was known about this. And then we started to look at which of the amino acids are phosphorylated of VP30, and we found out that it was mainly serines that were phosphorylated. So mainly we found phosphoserine when we analyzed the amino acids of VP30. And there is a cluster of serines in the N-terminus of VP30, 6 different serines, and we found by mutating the individual serine residues, or 3 of them together, that they were indeed the target for this unknown kinase, and they were phosphorylated because when we remove these serines, you can see that the phosphorylation signal is decreased or is more faint. And so we were sure that the phosphorylation could take place with all 6 serine residues. Then we took our mini replicon system and we asked whether the phosphorylation of VP30 was important for the transcription process. And you can see this is the wild type of VP30 that we used, and then we included a number of different mutants that were different in their phosphorylation status. So, for example, we had one mutant that had all serines. I don't know whether you can recognize this. Letters are too small. So we had one mutant where all 6 serine residues were replaced by alanines, meaning that this protein could no longer be phosphorylated, but the transcription activity was still present. And the only mutant which was not able to support transcription was a VP30 mutant where all the 6 serines were replaced by aspartate. And with this mutant, we had the serines all replaced by negative charged amino acids, simulating a phosphorylated serine at exactly this place. Meaning that in the presence of a phosphorylated VP30, transcription could no longer take place. So we needed at least a partially non-phosphorylated or fully non-phosphorylated VP30 to get this transcription active. And then we also checked whether the interaction between VP30 and the nucleoprotein inside of these inclusions that are formed by the nucleoprotein, whether they were also dependent on the phosphorylation of VP30. And as you can see here, in this panel, we had non-phosphorylated VP30, and you can see that there is a very weak interaction of VP30, which is here shown in a homogeneous distribution in this transfected cell, that there is an interaction with the inclusions formed by the nucleoprotein, but this is much more weak than in the presence of the phosphorylated VP30. Here you can see that VP30 and the nucleoprotein co-localized completely. So what we learned from this was that VP30 is phosphorylated at an N-terminal serine cluster. The phosphorylation of VP30 inhibited transcription, and the phosphorylation strengthened VP30's interaction with the nucleoprotein. And what we also learned from this set of experiments that I haven't, so the results I haven't shown you, was that VP30 could be dephosphorylated by cellular phosphatases PP1 and PP2A. Okay, then we were interested in the interaction of VP30 and VP35, whether this, on the one hand, whether these two proteins could really interact. And this was the case. So when we co-expressed VP30 and VP35, we could see that there is a co-immunoprecipitation. You can see here we pulled at VP35, and you can see that the VP30 flag tag was also co-precipitated. And this was also possible when we used the fully dephosphorylated VP30 mutant, then also the VP30 and VP35 could interact. But in the presence of the fully phosphorylated VP30 mutant, there was no interaction seen. And here you can see the quantification, and you see that there is a significant downregulation of the interaction between VP35 and the fully phosphorylated VP30. So VP30 interacts with the polymerase cofactor VP35 in a phosphorylation-dependent manner, and from this we conclude that there is also that VP30 also acts with the complex of VP35 and the polymerase during the viral life cycle. Then we were interested whether VP30, since it has a big influence on the transcription, whether VP30 could interact with RNA. And this was indeed the case, as you can see here with this electromobility shift assay. You can see that the wild-type VP30 was able to interact with an RNA which we synthesized from the first 154 nucleotides of the viral genome. This is the leader of the viral genome. And also the non-phosphorylated VP30, this mutant here, was able to interact with this RNA. However, when we used a fully phosphorylated VP30, or let's say a mutant that simulated a fully phosphorylated VP30, this was much weaker in its interaction with the genomic RNA. And then we were interested whether the RNA might be the factor that helped to form the complex between VP30 and VP35, which we found to be phosphorylation dependent in the experiment before. And what we found here was that in the presence of RNAs, so when we removed the RNA that bound to VP30, then the interaction between VP30 and VP35 was strongly reduced. And this here is the quantification of this experiment, meaning that most likely it's the binding to RNA which allows the binding to VP35. And when we remove the RNA, then the interaction between VP30 and VP35 is no longer possible. So on the one hand, VP30 is interacting with RNA in a phosphorylation-dependent manner, and what we showed before was that VP35 and VP30 is also, the interaction is also phosphorylation-dependent, but now it seems that this is all, the interaction comes via binding of the RNA, and if the RNA is depleted, then the interaction is no longer possible. So VP30's interaction with RNA is phosphorylation dependent, and the interaction with the polymerase cofactor VP35 is RNA dependent. Well, then we thought that phosphorylation of VP30 is always negative for the viral replication. But then we found that this is not always the case. When we looked with our mini-genome system in a setting where only primary transcription was possible, this is the step of the viral life cycle which takes place early after the virus entered the target cell, then we found that only in the presence of a serine at VP30, in the cluster of these serines of VP30, it was possible to see primary transcription. If we had no serine present in this VP30 mutant where all the serines were mutated to alanine, then primary transcription was no longer possible. And the serine at position 29 seemed to be a very important residue, because when we replaced all the other serines in the N-terminus of VP30, the background of a VP30 where all the other serines were alanine, then we could see that only serine 29 was able to rescue this effect. So serine 29 is important for on the one hand to be phosphorylated to rescue primary transcription. And this was very interesting to us, because we thought that a non-phosphorylated VP30 would do everything which was needed. But this experiment was also supported by rescuing experiments of recombinant Ebola virus, where we had this VP30 mutant, where we either had only serine 29 in the genome, in the genome of Ebola virus, serine 29 in the open reading frame of the VP30, or we had serine 30 in the open reading frame of VP30. And you can see, while rescuing was possible with serine 29, rescuing of serine 30 was not possible. And now, since this was an important amino acid, we generated an antibody that recognized specifically phosphorylated serine 29 to see how this phosphorylation status of VP30 is in the infected cell. So the conclusion of this experiment was that there is obviously a dynamic phosphorylation of VP30, which is essential for primary viral transcription and for the rescue of infectious recombinant Ebola virus. And it's important to have in mind that this is only true for primary viral transcription. As soon as you have a lot of viral proteins around, and so you are in the step of the secondary transcription, then you can deal with the fully dephosphorylated VP30. So this is then sufficient, but only for the primary viral transcription, you need this phosphorylated VP30. Okay, and then we, as I said, we had now an antibody with which we could see, we could detect and analyze the phosphorylation status of VP30. And then this was done in a transfection assay, and what you can see here, when we had VP35 and L co-synthesized with VP30, then there was the phosphorylation of VP30 seen. But as soon as we had the nucleoprotein co-synthesized with VP35 and L, then the phosphorylation of VP30 was decreased. So meaning that somehow the nucleoprotein was necessary to allow the dephosphorylation of VP30. This was very interesting. And then we had a collaboration with Jakob Nilsson from Copenhagen, and they have identified the binding site on the nucleoprotein for B56, and B56 is a subunit of PP2A. And as you maybe remember, we found some years ago that PP2A is a phosphatase that dephosphorylates VP30. So this phosphatase binding site on the nucleoprotein is very close to the VP30 binding site on the nucleoprotein. And so the idea was now that the phosphatase binding and VP30 simultaneously, the VP30 binding is important, that VP30 can be dephosphorylated by PP2A. And this hypothesis was supported by the fact that when we removed the PP2A binding site here, then the phosphorylation of VP30 was increased. And so meaning that if the phosphatase can no longer bind here, then VP30 stays in its phosphorylated status. And this was also supported by another set of experiments when we removed the VP30, when we removed again the PP2A binding site, that this strengthened the phosphorylation of VP30, as you can see here. So this is the wild type VP30 and this is the mutant VP30 with the B56 or the phosphatase binding site removed. And you can see that this phosphorylation then is increased when you compare this to a nucleoprotein where the phosphatase binding site was present. There, the phosphorylation of VP30 is very much decreased. So, sorry, it's difficult to read this here. So NP recruits the phosphatase PP2A by recruiting the subunit B56 in order to dephosphorylate VP30, and this then activates the transcription of Ebola virus. Okay, then the last piece of this puzzle to get a fully dephosphorylation circuit of VP30 was to identify the kinase that recognizes VP30. And for this, we did proteome analysis, or let's say a mass spec analysis with VP30, and we identified binders, cellular proteins that bound to VP30. And there, one kinase came up, and this was SRPK1. And SRPK1 is a kinase that is necessary for splicing of mRNA in the nucleus of the cell. But SRPK1 was also able to bind to VP30. And we also could in vitro phosphorylate VP30 with SRPK1, as you can see here when we looked at the phosphorylation status of VP30 that was incubated with SRPK1. And you can see that the mutant of VP30, either the non-phosphorylated or fully phosphorylated, simulating VP30, that this could no longer be recognized by SRPK1, but the mutant where only the serine 29 was present could be recognized by SRPK1 and phosphorylated. So, and we also identified then the SRPK1 binding site on VP30, and when we tried to rescue a virus that where this binding site was mutated, you can see that this virus was much impacted in its growth because this mutant could no longer be recognized by SRPK1. So VP30 is phosphorylated by SRPK1 via an RXXS recognition motif in the N-terminal to the serine cluster of VP30. So SRPK1 directly binds to VP30 and phosphorylates then here serines from this N-terminal cluster. Okay, now I talked a lot about these different results that we had with VP30, the phosphorylation, dephosphorylation, phosphatase binding, and kinase binding, and so on. And now I would like to wrap this all together into one working model. What you can see here is a cryo-EM structure of one subunit of the nucleocapsid. So here we see the nucleoprotein, the N-terminus of the nucleoprotein. There we have a crystal structure. Then we have a crystal structure of VP24, which is also bound to the nucleocapsid. And then what we see here is that there is a dimer of VP24 bound to this to the nucleoprotein. And we can see here in the structure shown in the cryo-EM, but not in the crystals yet, which is the C-terminus of the nucleoprotein, and most likely also the structure of the VP35. Okay, this is here the C-terminus of the nucleoprotein, and here you see the binding sites for the phosphatase 2A, and you see the binding sites of VP30 on the nucleoprotein. So the idea is now that first of all, the phosphorylated VP30 which can very strongly VP30 binding site, and the phosphatase is able to dephosphorylate VP30, which weakens the interaction with the nucleoprotein. And then VP30 can be relocalized to the RNA, to the genomic RNA, and especially to sites where the RNA forms a strong secondary structure. And then this binding of VP30 to the RNA can then recruit the polymerase, which binds to this complex and then starts to form the mRNA, the transcription. So with this, I'm already at the end. I will skip the second part of my talk because I'm running out of time, and just wanted to stop this and go to the last acknowledgement slide, which is almost the most important thing. So the work that I showed you today was mainly about the VP30 phosphorylation and dephosphorylation. And there, the main actor was Nadine Biedenkopf, who did all this work during the last 10 years, together with work that has been done previously before Nadine joined the lab by these people here, especially Elke Mühlberger was important, Viktor Brechot and Thomas Höhn. And the second part that I didn't have the chance to show you today was about VP40 of Ebola virus, which has several redox modifications.
Maybe you can, Stephan, if you want to show 2 or 3 slides on this.
Yeah, okay. I can, if you allow. Now we are 3:45.
Yeah. If you just show 2 or 3 slides to have at least a feel of your results.
Okay. So this part is about VP40, and VP40 is the budding, the matrix protein of Ebola virus, which induces budding at the plasma membrane. And VP40 comes along with several homooligomerization states. It starts with a dimer of VP40. And this dimer of but not only the dimer is important, but there's also a hexamer or a filamentous form of VP40 and a circular octameric form of VP40. Now, the dimer can be transported to the plasma membrane, where it forms hexamers and filaments finally. And these filaments are then important for the budding process of the viral particle. On the other hand, VP40, can form these octamers, and these octamers are important for an inhibition of the viral replication and transcription. So we were looking at this at VP40 in another project where we tried to make a structure-based drug design for antiviral against VP40. And Anke Werner, she was able to show that there is she got a high resolution structure of VP40, and she was able the first time to see the C terminus of VP40 in the crystal structure. And the C terminus of VP40 has 2 cysteines, and what she could show was that these cysteines formed a disulfide bridge, as you can see here. So this was interesting for us because we thought, well, if there is a disulfide bridge, maybe the cysteines are also necessary for some functions of VP40. And therefore, we looked at these 2. Here you can see again the C-terminal end of Sudan VP40, and you can see these 2 cysteines can form a disulfide cluster. And then we try to characterize this cysteines by cysteine to serine mutants in functional and biochemical assays. And now I skip this and I show you here that in the mini-genome assay, when we used mutations of VP40, which had instead of the cysteine a serine, then you could see that the wild type was able to inhibit transcription of the mini-genome, but the mutants of the cysteine mutants of VP40 were no longer able to do this. But to our surprise, the double mutant was as good as the wild type in inhibiting the mini-genome activity. So this was interesting. This was the same for Zaire VP40. And Then we looked at the other function of VP40, at the budding function with this mutants of the cysteine mutants of VP40. And what you can see here is that the filament length of these protrusions at the plasma membrane became longer in the presence of these mutants of VP40. And you can see electron micrographs that show the same bit, that that the mutant of VP40 has much longer filaments at the plasma and uses much longer filaments at the plasma membrane than the wild type, as you can see here. Okay, then Anke tried to rescue these cysteine mutations in VP40 in a recombinant virus. And what you can see is that this was possible, but the virions, the recombinant variants, were slower in the growth than wild type. And they also induced longer filaments at the plasma membrane. And what was most interesting, the particle length of the released virions was longer with the mutants than with the wild type. So then finally we investigated the redox modification of the cysteines at the C-terminus of VP40 with a biotin switch assay, and we found that indeed the cysteines could be biotinylated, which means that these are nitrosylated, which is checked with this biotin switch assay. And then with mass spec analysis, we could also show that the cysteines were not only nitrosylated, but also glutathionesylated, sulfonylated, and sulfidylated. I don't know how to pronounce this. And now the question which is open so last point. So when we looked at these redox modifications, we could also show that with a thioredoxine assay, that the VP40 of Sudan Ebola virus, but also Zaire Ebola virus, is the direct target of thioredoxine, which is then which needs thioredoxin reductase for its reduction again. So finally, what we don't know currently is whether it's the redox modification of the cysteines or the cysteines themselves that makes the phenotype of these mutants, and maybe and probably it's both, that we need the redox modification and the cysteines. Okay, with this I stop here, and again, these are the acknowledgements. So the work with VP30 was also had the help of Jakob Nielsen, and we had the help of Erika Allman Sapphire for Anke to learn how to crystallize VP40. Thank you very much.
Thank you so much, Stephan. Very elegant lectures and a very good example of how much we need very basic molecular virology and to really get the new concepts and on these viruses. Very elegant. So we have some questions of the Q&A. We can start with them. I guess you can see them. You can read them, Stephan.
I can try, yes.
Otherwise, we can.
No, I have it. So the first question is by Jelena Kalle. Which methods were used to obtain mutants? So we used simply in vitro mutagenesis to induce the mutants of, if we replace cysteines by serines or serines by alanines in case of VP30. And then it's Stefan Ludwig. Hi, Stefan. Good to hear you here. Thank you for your talk. VP30 phosphorylation. How about proper timing of phosphorylation and especially dephosphorylation? Is dephosphorylation just regulated by increasing presence of NP and PP2A recruitment, or is there another layer of phosphatase regulation? Thank you, Stefan. And Stefan Ludwig, it's necessary to mention he is an expert in phosphorylation and dephosphorylation of viral proteins. So what happens is that the VP30, which can directly interact with the kinase, this SRPK1, is then recruited as a phosphorylated protein into the inclusions, and then binds very tightly to this NP-VP30 interaction site. And then this is in close distance to the PP2A binding site. And then the PP2A can dephosphorylate VP30, and then VP30, this weakens the interaction of VP30 with the nucleoprotein, and then the VP30 can bind to the RNA. So at least this is so far what we know about, and whether there is another layer of phosphatase regulation, we don't know. And then there is a question by Anders Bahn. I don't know, Christian, should I proceed?
Yes,
with
please, absolutely.
The virion VP30 is dephosphorylated only partially. What we know is that at least in the virion there is phosphorylated VP30, but we think there is also dephosphorylated VP30.
Then you have a question from Bob Gary.
Yeah, thank you very much. Thoughts about how Ebola virus might occasionally go dormant and then reactivate in some survivors. Bob, this is a $100,000 question, which is very difficult to answer. Better to ask Gustavo Palacios, I guess, than me. So actually nobody knows exactly. So we see Ebola virus on the one hand in the semen or in the testis of the yes, thanks. We see them in the semen or in the testis of the Ebola virus survivors. And what we also know is that the mutation, as long as Ebola virus is dormant in the survivors, that the mutation rate is very, very slow, meaning that most likely the replication, the amount of virus that is replicated is probably not very, very high, meaning that there is a very low, my suggestion would be that there is a very low replication rate of Ebola virus in the testis. This maybe allows that the virus can survive there without being recognized by the immune system. But actually, I don't know. This is the short answer.
Thank you. Anybody who has an idea is most welcome on this question. Okay, well, I have another question, which is obvious also a bit, but with such a fine description of the overall replication mechanism, post-translational, I mean, obviously you are looking for targets for new molecules, for new drugs. Do you have any insight as to how what you have been doing can really lead to some new targets, maybe you cannot speak in detail, but do you have collaborations on this? How does it go?
I mean, it's as you said, it's obvious to look at, for example, the interaction between VP30 and the phosphatase to somehow inhibit the dephosphorylation of VP30, which would finally then inhibit the replication or transcription of the whole process. And this is in principle a good idea, but PP2A is an enzyme that is needed for numerous cellular processes. And if you inhibit this phosphatase, it's highly cytotoxic for the cells. And therefore, it's always difficult to play around with this kinases and phosphatases. Although there are a lot of kinases which are drug targets already, and it might be a good idea to look at kinase inhibitors that we know are also important for the transcription of VP30. But
sure. Thank you very much. I do not see other questions. No? Well, thank you so much, Stephan. And we really hope that you will be contributing to several of our activities in the GVN, and congratulations for this seminar. That was really great. Thank you so much, and thank you everybody for attending.
Thank you very much, Christian, and thanks to everybody. Bye-bye.